Welcome to the lecture on Genetic Engineering Part 1. This is part of the Agricultural Technologies subject ALM207, which is offered at NMIT as part of the agricultural degree. My name is Dr. Nikki Cooley. The lecture on genetic engineering today is part of the topic Technology in Plant and Animal Breeding. This topic starts with an introduction to genetics, followed by looking at some aspects of plant and animal genetics. Please ensure that you watch these lectures before continuing. In Lecture 1 we are going to cover the details and descriptions of these technologies and spend some time understanding the terminology so that we can understand these technologies. Lecture 2 will be structured slightly different, in that there will be a presentation of technologies in the form of case studies and exercises for you to complete as you go through the lecture. Please watch the following video made by the Open University. This video describes or gives an introduction to genetic engineering and particularly bacteria, where much of this technology started from. Please note that you do not have to learn the contents of this video for assessment, but it does set the scene for GMOs and gives a very good introduction of this technology. Now I would like you to watch a second video. This video is on genetic code and agriculture. This video puts the role of GMOs in perspective of the traditional breeding methods and gives you some examples of some genetically modified crops, and also gives you some visualizations of this method. Now we have an overview of GMO and genetic engineering. Let's get on top of some of the definitions and explain and define these. Let's start with a definition of transgenetics. This is the process of introducing an exogenous gene, called a transgene, into a living organism, so that that organism will exhibit a new property and transmit that property to its offspring. When an organism has undergone transgenic modification, it is often referred to as genetically modified organism, or as a GMO for short. There are now many methods to facilitate the transfer of these genes. Here we will describe an overview of some of these methods, but please note that this list is not complete. Plasmid vectors. A plasmid is a small DNA molecule that is physically separate from and can replicate independently of chromosomal DNA within a cell. Vectors are an arrangement of genetic material and express particular genes or make copies of these genes. Liposomes. A liposome is an artificially prepared vessel composed of a lipid bilayer. Viral vectors. Viruses are able to transfer genetic material into the cells. Pronuclear injection. This injection of genetic material into the nucleus of a fertilized oocyte, female gameocyte or germ cell. Protoplast, or more commonly known as stomatic fusion. This is genetic modification in plants by which two distinct species of plants are fused together to form a new hybrid plant, with the characteristic of both, a stomatic hybrid. And finally, ballistic DNA injection, a device for injecting cells with genetic information. So, how does transgenics work? Well, the concepts are that all living organisms carry the same form of genetic code, as we know DNA, and this is made up of small building blocks in organisms. These are base pairs A, C, T and G. And the processes behind this was covered extensively in the previous lectures. This means that a specific DNA sequence will code for the same protein in all organisms. The transgenics in principle is a piece of DNA is cut from one organism and put into the other organism. The figure on the screen describes visually the differences between conventional breeding and transgenics. Here we have a simple concept of using transgenics in animals. So we start with creating a transgenic animal. DNA of choice, that is, DNA that exhibits a trait that you wish for your animal to exhibit. This is identified and prepared in a laboratory scenario. Once the DNA with your transgene or gene of choice is prepared, it is then injected into the egg of the animal that you wish to breed with. The egg is then implemented into a surrogate cow, in this case. The offspring will then demonstrate this trait. Let us look at one example of a technique of transgenics in plants. This is known as the agrobacterium technique. Put simply, a trait of interest is identified. The tDNA is then inserted into a plasmid. This plasmid is placed into the agrobacterium. The agrobacterium is able to insert this genetic code into our plant cell. The plant cell reproduces and generates into a new plant. This new plant and its offspring will generate or illustrate the new trait. In the scientific articles, you can now read about some technologies that are being developed. These include the zinc finger nucleases, the transcription activator-like element nucleases, and thirdly, a new delivery system by agroinoculation using different specialized vector systems. All of these technologies are being developed in plant genetic engineering. We will not cover these in any detail, as they are in the very early stages of development and not yet available for commercialization. This does, however, give you an idea for how rapidly this field of technology is developing. Molecular biology plays an essential role in all breeding technologies. Here are a couple of examples of what is called high-throughput DNA marker platforms. This means that many DNA sequences can be identified at once or in one run. These technologies include SNPs, microsatellites, AFLPs, and mobile genetic elements. In the past, the identification of one or two genes was conducted at a time. This has been very expensive and time-consuming. However, technology is gaining pace, and now a new suite of technologies exists so that we can identify many genes or even the whole genome at once. This field is called the science of genomics, or genomics. When we were able to identify the whole genome-wide gene expression, we get an output similar to the left-hand side figure on the screen. This means that we can identify which genes are upregulated, which genes are downregulated, and which genes are not switched on or off. This gives us an amazing amount of data which we can then correlate to certain traits. The process of taking all of this information into the mathematics behind identifying how this information integrates from expression patterns to the observed traits is very complex. It is developed using Bayesian machine learning strategies. As well as the technological advances in the statistical and mathematical processing of gene traits. The other very important criteria for allowing this technology to become commercially available and develop is its falling cost. For example, between 1990 and 2003, it cost about $13 million to sequence genes. That has fallen from the 2012 cost of only $100 to sequence a whole genome, and it takes less than 10 minutes, which is an impressive achievement by technology and scientists. So what are some of the technological difficulties in the GM technology? Well, one such limitation is the DNA addition only, so no DNA replacement. This is still far from gene targeting in crop plants. Secondly, there is no targeted integration in the genome, with possible unintended effects on the plant phenotype. The EU risk assessment has shown that this needs to be addressed. 3. Non-targeted integration with unpredictable effects of transgenome expression or positive effects. Agronomic testing and EU risk assessment procedures are addressing this point. 4. There is a low efficiency in transformations which may require selection markers or antibiotics resistance, raising safety concerns. Alternatives to the use and the persistence of antibiotic-resistant marker genes in the marketplace of GPMs exist. Extensive risk assessment failed to identify any adverse effects until now. One of the very important components of GMOs and risks associated with it is containing the traits or genetics that you do not want to get out into the environment. This was called controlling gene flow. The molecular farming needs containment. There is little discussion or there is agreement in the scientific community that this is true. Biological containment is an option. This may be achieved by several methods. There is male sterility, seed sterility, cleostomography, inducible promoters of transgene expression, transgenic mitigation, and maternal inheritance. It was said by Johnson and Dolmore in 2002, by choosing the right plant, the development of gene containment mechanisms may be unnecessary. On the screen there is a web-based link to a YouTube video, which is a report documented by the BBC. This report is on genetic engineering and potato crops, science and application in the UK. Please answer the following questions. Question 1. What was the problem the GMO technology was trying to address? Question 2. How were GMOs correct for this? Question 3. How do the scientists produce GMO potatoes? Question 4. How much money has been spent on the project being discussed in this video? Question 5. What are the economic impacts for UK farmers? And question 6. What does Patrick Holden, the Director of Soil Association, think is the problem with this technology? Please pause the video until you have had a chance to answer all these questions. I will now go and attempt to answer these questions. This is a learning example of how you answer these questions with information obtained for video. The problem with the GM technology was trying to address, as described in the ABC report, was the problem of potato blight. This is a significant problem in the farming of potatoes. It requires 15 times more sprays a season when potato blight is treated by traditional methods. The GMOs will correct for this by inserting new genes into the existing crop. This will ensure less spraying of harmful chemicals is required. The scientists produce GMO potato crops in the following way. 1. They use wild potatoes. 2. They identify the traits that exhibit resistance in these wild potatoes. 3. They then sequence the resistant traits. 4. They insert the sequence of the resistant traits into the modern varieties and enable disease resistance while keeping the modern varieties' characteristics. There is about £1 million spent on this project. The problem of potato blight costs UK farmers losses of £16 million a year. Patrick Holden, the Director of the Soil Association, thinks there is significant problems, as there is not only loss of public funds, but these are not producing public benefits. No farmer benefits, there are threats to the environment, and this technology is potentially harmful to the health, are all arguments that are put forward in this video. I want to end from a summary of some observations taken from a gene slicing paper shown on your screen. This paper was written with respect to gene silencing and plant genetics. However, these characteristics can be taken across all genetic engineering. The authors concluded that agronomic traits are controlled by genes, but they are also controlled by gene networks and by heritable and non-heritable gene regulators. This means that there are some regulators of genes which are transferred from one generation to the next, while there are other non-heritable gene regulators. That means they're not transferred to the next generation. 2. Empirical selection, which is based on phenotype, is supported by genetic and epigenetic determinants of emerging complexity. And 3. Gene regulators, not only gene combinations, must be captured by plant breeding and by selection of plant varieties. That brings us to the end of part 1 of this lecture. In summary, we have introduced the concepts of genetic engineering, both within this lecture and in the plant and animal breeding lectures. We have given a description of genetic engineering and transgenics. Some terminology in this field is described. Examples of 2 transgenic methods used in both plants and animals are described. We have looked at the development techniques and where the future may lie. We have touched on the introduction to genomics and the role it plays in the production of GMOs. We have looked at an example of genetic engineering of potatoes and the pros and cons of this technology. We've concluded this lecture with some take-home messages about genetic engineering technologies.